Hello guys, welcome to my channel. So today we'll study about the disaccharides. So in the previous videos we studied out about the monosaccharides. So now in the carbohydrates we'll study about the disaccharides. So we'll study about the disaccharides. So disaccharide, so such as maltose, lactose, sucrose, right? So they are maltose, sucrose, lactose. These are all disaccharides, right? Why disaccharides? So two monosaccharides joining covalently by an O-glycosidic bond. So two monosaccharide join by joined covalently by O-glycosidic bond. So two monosaccharides joined by O-glycosidic bond. What does that mean? All right, so I will show you what does this mean. For example, this is alpha glucose, and so this is one monosaccharide, right? And this will join again and this is, for example, beta glucose, right? And this is hemiacetal, if you remember that we studied in the previous video when we studied about the monosaccharide. So this is hemiacetal, and this part is an alcohol, right? COOH, that's an alcohol, right? So when, okay, so two monosaccharides join covalently by an O-glycosidic bond, which means that when a hydroxyl group, so this is the hydroxyl group of one sugar molecule, typically in its cyclic form, so this is a cyclic form, right, reacts with the anomeric carbon of the other. Reacts with the anomeric carbon of the other. So what does that mean? Anomeric carbon is this carbon atom. If you remember, we studied about anomers, right? Right, we studied about the anomeric carbon atom. So for example, this carbon atom is an anomeric carbon atom here, the first carbon atom, right? So I will just putting images here to just represent or to refresh your memories regarding the anomeric carbon atom. As you can see in the image, the alpha D glucopyranose, that's an anomeric carbon atom. But when it's fructofuranoose that we studied about later, the C2 is an anomeric carbon atom, as in ketose, right? If it's a straight chain, you remember that's in the bottom, that's aldehyde and the tail end of the, right? And if it's from C2 to C5, it's a ketone, right? Or a ketose. So if it's C2, it's a anomeric carbon for the fructofuranoase. All right, so what happens next? I'll show you. So with the hydrolysis or condensation reaction, okay? So from here, this actually join, instead the hydrolysis will join the oxygen here with the second here. Okay, so let me do this here. All right, so what happens now? Right, so what happens now? So this reaction that is shows represent the formation of acetal from a hemiacetal, such as a glucopyranose and an alcohol. What does that even mean? Right, so the disaccharide is formed from the two monosaccharide, correct? The two D-glucose, so this is alpha D-glucose actually molecule, both are, and beta D-glucose molecules. All right, so when an hydroxyl group of one monosaccharide condenses with the intramolecular hemiacetal, right, hemiacetal of the other with the elimination of H2O, that's an hydrolysis, right? So when it's hydrolysis, H2O, this direction, H2O, right? Oops, so I did the wrong thing. This go up and this go down, right? Okay, so the elimination of H2O, right, with the elimination of H2O and the formation of the glycosidic bond. So this is the glycosidic bond. So the reversal reaction is this hydrolysis, right? The condensation reaction and hydrolysis that is attacked by an H2O on the glycosidic bond. So when the disaccharide is formed, when the two monosaccharides that you can see here, when an hydroxyl group of one monosaccharide, that is an acetal, condenses, that's make a condensation reaction with the intramolecular hemiacetal of the other with the elimination of H2O, it forms a glycosidic bond, right? So this is acetal, this become from hemiacetal to acetal, and this is hemiacetal now. This side, and the reversal reaction, the hydrolysis, right, is attacked by the H2O. This is attacked by the H2O on the glycosidic bond here, the maltose, and this molecule is, what's the disaccharide? It is maltose, right? This maltose molecule retains a reducing hemiacetal. So this is a reducing hemiacetal at the C1. And okay, so I will number the carbon atoms for you. So for example, this is 5, this is 6, right? This is 4, this is 3, this is 2, and this is 1. So this is 1, the same here, right? The same numbering of the carbon atom. So the maltose is a reducing retains a reducing hemiacetal at the number 1 position, that is not involved in the glycosidic bond. So this is not involved in the glycosidic bond, but it's a separate one, because the muta rotation interconverts the alpha and the beta forms of the hemiacetal. It converts so the bond at this position are sometimes depicted with the wavy line to indicate that the structure may be either alpha or beta, right? All right, so this reaction represents the formation of acetal from a hemiacetal, right, such as glucopyranose. So this is the glucopyranose actually, hemiacetal, and the alcohol, that is this one, right, and the resulting compound is called a glycoside as well. So the glycosidic bond, this is formed, are readily hydrolyzed, right, by acid, but resist cleavage by base. So this can split easily by an acid. Split easily by acid. Okay, this can split easily by an acid, right? So thus disaccharide can be hydrolyzed to yield their free mode. So this can be hydrolyzed to yield free monosaccharide components by boiling with the dilute acid, and the N-glycosyl bonds join the anomeric carbon of a sugar to a nitrogen atoms in glycoproteins and nucleotides. So that we'll study when we'll study about the glycoconjugate. But remember this bond, the N-glycosyl bond, and that's an anomeric carbon atom, as we studied what are anomers, right? So the oxidation of sugar by cupric ion, that the reaction that defines a reducing sugar, occurs only with a linear form which exists in an equilibrium with the cyclic forms as well, right? So the oxidation of sugar by a cupric ion, so that exists occur only in the with the linear form, but which exist in equilibrium with cyclic forms as well. So when the anomeric carbon, so these are the anomeric carbon, right? Number one, uh, in a glycosidic bond, so this are the anomeric in the glycosidic bond, that is when the compound is fully acetal or kettle. The easy interconversion of linear and cyclic form is prevented. So what does that mean? So when the anomeric carbon, so these are the anomeric carbons, right? Anomeric carbon is involved in a glycosidic bond, that is when the compound is fully acetal. This is not fully acetal. This is acetal and this is hemiacetal. But when it is like this is acetal and this is also acetal, or this is kettle and this is also kettle, the easy interconversion of linear and cyclic form is prevented. Why? Because the carbonyl carbon can be oxidized only when the sugar is in its linear form. That is, formation of a glycosidic bond renders a sugar non-reducing. So in describing disaccharides or polysaccharides, the end of a chain with the free anomeric carbon is commonly called the reducing end. Right? So when the anomeric carbon is involved in a glycosidic bond, like if there is a glycosidic bond one here as well, the sugar will be non-reducing. But as here you can see this anomeric carbon, or when describing disaccharide or polysaccharide, the end of a chain with a free anomeric carbon that is not involved in a glycosidic bond is commonly called reducing end. So this will known as a reducing end, correct? So that means the disaccharide maltose, right, contains D-glucose residue. That D-glucose residue that is joined by a glycosidic linkage between the carbon 1, the anomeric carbon of one glucose residue, and the C4, that is the 4 number right here, the C4 of the other. And because the disaccharide retains a free anomeric carbon, maltose is a reducing sugar. So this means that is it is a reducing sugar, right? And the configuration of anomeric carbon atom in the glycosidic linkage is alpha. So the glucose residues with the free anomeric carbon is capable of existing in alpha and the beta pyranose form. And we'll see now the naming, how we name. So for example, this maltose will be named like this, the glucopyranosyl with carbon, that is 1, 2, 4, with D glucopyranose. So how this is named like this? So we'll see. In order to name any reducing disaccharide such as this maltose, or like especially to name more complex oligosaccharide, there are several rules to be followed. So by convention, this name describes the compound written with its non-reducing end to the left. So from here, right, and we can build up the name in the following order. So first, the given configuration, alpha or beta, at the anomeric carbon, right here, okay, anomeric carbon, joining the first monosaccharide unit on the left to the second, right? So this is on the left, that is the second one, right, from the anomeric carbon. So we'll just put alpha or beta here at the first, right? Name the non-reducing residue to distinguish 5 and 6 member. So as number 1, so we'll give the configuration, the name, whatever, alpha or beta, and the D-gluco for this part, alpha D-gluco for the first part. And the second, the name non-reducing residue to distinguish 5 or 6 membered ring structure. So if it's 5 or 6 member ring structure, we can insert furano or pyrano into the name, furano or pyranos, as you can see. And third, indicate in the parenthesis that the two carbon atoms, this two carbon atom joined by the glycosidic bond with an arrow. So we'll show here the two carbon atom. So this is number 1 and this is number 4. So that's why it says 1, 2, 4. Shows that the carbon 1 of the first named sugar residue is joined with the carbon 2 at the C4, right? Then the second residue, this one, right? If there is a third residue, describe the second glycosidic bond by the same convention. So it goes on and on the same way, right? And like when we describe polysaccharide in the name, so just to shorten the name of the complex polysaccharide, there is like 3-letter abbreviations or color symbols for the monosaccharides are often used. And following this convention for naming oligosaccharide, maltose is alpha D-glucopyranosyl 1,2,4 and D-glucopyranose, because most sugar encountered here are the D-enantiomer, right? And the pyranose form of hexoses predominates, right? Right, and on your screen you will see some symbols and abbreviation of common monosaccharides and some of their derivatives. That's the table, and this is how it is written, the symbols and the abbreviation. All right, in the same way, maltose, that is the disaccharide lactose. I'm not drawing the structure for the lactose. You can see on your screen the lactose. So this is how the lactose is formed, which yields D-galactose and D-glucose on hydrolysis. When the lactose, for example, like this is hydrolyzed, it yields D-galactose and D-glucose that occurs naturally in the milk, right? And the anomeric carbon, right? The anomeric carbon of the glucose residue is available for oxidation, and thus the same here, like as an the anomeric carbon is available for oxidation, and thus lactose is as well as a reducing sugar or reducing disaccharide, and its abbreviated name is, as you can see, GAL. It's beta 1 2 4, right? GLC. So the enzyme lactase, absent in the lactose intolerant individuals, right? The enzyme lactase in the individual, when in humans, like for example it's me or it's you. So when the enzyme lactase is absent in whoever is lactose intolerance, that begins the digestive process in the small intestine by splitting this beta 1 to 4. So this is the where it splits, right? Bond of the lactose into monosaccharides, which can be absorbed from the small intestine. But lactose, like other disaccharide, is not absorbed from the small intestine, and in lactose intolerance individuals, the undigested lactose passes into the large intestine. So in the enzyme lactase, that is absent in the lactose intolerance individual. So what happens? That begins the digestive process in the small intestine by splitting the carbon 1 and the 4, as it's named as beta 1 to 4, right? The bond of lactose into monosaccharides, which can be absorbed from the small intestine. But lactose, like other disaccharide, is not absorbed from the small intestine. And in lactose intolerance individual, what happens? The undigested lactose, because there is no enzyme lactase, that passes into from the small intestine into the large intestine, and here the increased osmolarity due to dissolved lactose opposes the absorption of water, right? Because of the lactose, the water is not absorbed from the intestine and into the bloodstream, and that causes a watery loose stools, right? And in addition, fermentation of lactose by intestinal bacteria produces large volume of carbon dioxide, which that also leads to bloating, cramps, and gas associated with the lactose intolerance. And this is how if you're lactose intolerant and if you drink milk, this is the symptoms you get because of the fermentation or because of the large volume of carbon dioxide and increased osmolarity, right? All right, and same as lactose and maltose, that is sucrose. Sucrose is also a disaccharide of glucose and fructose, and it is formed by plants but not by animals. And in contrast to maltose and lactose that we saw, that it has a free anomeric carbon, or it is a reducing sugar, but sucrose contains no free anomeric carbon atom. So the anomeric carbons of both monosaccharide units, like this, for example, if this is a glucose and fructose, the both anomeric carbon atoms, right, of the monosaccharide are involved in the glycosidic bond. Right, as you can see on your screen, this is the structure of sucrose, and this is how the both anomeric carbon atoms are involved in the glycosidic bond. So sucrose is therefore a non-reducing sugar, and that's why its stability, its resistance to oxidation makes it a suitable molecule for the storage and transport of energy in plants. And in the abbreviated nomenclature, a double-headed arrow connects the symbols specifying the anomeric carbon and their configuration. So that's the why the abbreviated name of sugar is that you can see on your screen. This is the abbreviation as we did in the maltose, right? And sugar is a major intermediate products of photosynthesis, and in many plants it is a principal form in which sugar is transported from leaves to other parts of the plant body. All right, so there are like sucrose, and there are like three halos as well, a disaccharide of D-glucose. That's a disaccharide of D-glucose that, like sucrose, is a non-reducing sugar and is a major constituent of circulating fluid hemolymph of insects. It serves as an energy storage compound. All right, and lactose gives milk its sweetness. So why the milk are sweet? Because of lactose. Sucrose, of course, is a table sugar, as we know. Trehalose is also used commercially as a sweetener, right? The trehalose, as I mentioned earlier, that is a disaccharide of D-glucose, and that, like sucrose, is a non-reducing sugar, and that is a major constituent of circulating fluid of insects. But that trehalose is also used as a commercially as a sweetener, right? Okay. All right, guys, so that was disaccharide. So we finished monosaccharide earlier. Today we finished disaccharide. So, all right, so that's the description of disaccharide. All right, guys, so we'll see you in the next video. Thank you guys for watching, and don't forget to subscribe.